The first big proof of concept came in 2015 from researchers like James Kirkland and Judith Kempisi.
What did they find?
They found that a combination of two existing compounds, dasatinib, a cancer drug, and quercetin, a natural flavonoid found in plants, could selectively kill senescent cells in mice.
And did it help the mice?
It did. The mice showed improved physical function, reduced age-related pathologies, and even lived longer. It was a landmark study.
Has this moved into humans yet?
Yes, early-stage human trials are underway and showing some promising results. For instance, a 2019 study gave that D plus Q combo to patients with idiopathic pulmonary fibrosis, a fatal lung disease linked to senescence. The patients showed improved mobility, like how far they could walk in 6 minutes, and reductions in senescence markers in their blood. Other trials are looking at kidney disease, Alzheimer's, osteoarthritis. It's a very active area.
And you don't have to take these drugs constantly.
That seems to be a key feature. Because senescent cells accumulate relatively slowly, senolytic therapies might only need to be given intermittently, maybe just for a few days, every few weeks or months, to have a lasting benefit by clearing out the existing burden.
Okay, clearing zombie cells is one thing. What about going right into the source code? Directly manipulating our genes with tools like CRISPR and prime editing?
Yeah, this is arguably one of the most powerful and maybe ethically complex frontiers. Our DNA is the blueprint, right? And errors, mutations, damage accumulating in that blueprint definitely contributes to aging and disease.
So CRISPR is like molecular scissors.
That's the common analogy, yes.
Yeah.
CRISPR-Cas9, which won the Nobel Prize in 2020 for Charpentier and Dudina, was adapted from a natural defense system in bacteria. It uses a guide RNA to find a specific spot in the DNA, and the Cas9 enzyme makes a cut, usually across both strands.
And then the cell tries to repair that cut.
Right. And scientists can hijack that repair process to, for example, disable a faulty gene or sometimes insert a new piece of DNA.
What are the longevity uses?
Obvious targets are rare genetic diseases that cause accelerated aging, like progeria, but also potentially deactivating genes involved in cancer risk, modifying genes that control cholesterol, maybe even fixing mitochondrial defects.
But CRISPR isn't perfect, right? It cuts, but the repair can be messy.
That's been a limitation. It's great at breaking the gene, but controlling precisely how it gets put back together is harder. That can lead to unintended edits or off-target effects.
And that's where prime editing comes in, the upgrade.
Exactly. Developed by David Liu's lab in 2019, prime editing is often called search and replace gene editing. It's more sophisticated.
How does it work differently?
It uses a modified Cas enzyme fused to another enzyme called reverse transcriptase. It still finds the target DNA site, but instead of making a double-strand break, it typically just nicks one strand. Then it uses an RNA template to directly write a new genetic sequence into that spot.
Like using find and replace in a word processor.
Precisely. It allows for targeted insertions, deletions, or swapping single DNA letters with much higher precision and potentially fewer unintended consequences compared to traditional CRISPR-Cas9.
So much more accurate for fine-tuning.
That's the promise. Critical for aging research, where you want subtle adjustments, not wholesale disruption. And the applications are already emerging. In 2021, a study used a CRISPR-based editor in mice to correct the mutation causing progeria.
And did it work?
It extended their lifespan nearly 2.5 times. It was remarkable. For heart disease, a company called Verve Therapeutics is using base editing, another CRISPR variant, to permanently disable the PCSK9 gene in the liver.
To lower bad cholesterol.
Exactly. Permanently, with potentially single treatment. Looking further out, these tools might enhance our natural DNA repair pathways or rejuvenate the immune system by editing immune cells.
Of course, this raises huge ethical questions. Designer longevity.
Absolutely. The potential for misuse, unintended long-term effects, equitable access. These are massive societal discussions we need to have. And you even have DIY biohackers experimenting with CRISPR kits at home, which adds another layer of complexity and risk.
Incredible potential, but definitely needs careful navigation. Okay, moving from the DNA code to the energy factories, the mitochondria. You mentioned mitochondrial dysfunction as a hallmark. How are we tackling repairs there?
Right. Mitochondria were crucial. They produce ATP, the energy currency for basically everything our cells do. As we age, they tend to become less efficient, generate more damaging free radicals, oxidative stress, and their own DNA can accumulate mutations.
Leading to fatigue, metabolic issues, neurodegeneration.
Exactly. It's implicated in a huge range of age-related problems. So boosting mitochondrial health is a major longevity target.
How do we do that?
Well, some strategies focus on supporting them. Boosting levels of NAD plus IgE, using precursors like NMN or NR, is key because NAD plus is vital for mitochondrial function. Certain antioxidants, like MitoQ, are designed to specifically target and protect mitochondria from oxidative damage.
Supporting the existing machinery. What about fixing broken parts?
That's where it gets more futuristic. One really clever idea is allotopic expression. Mitochondria actually have their own small loop of DNA, mtDNA, separate from the main DNA in the nucleus.
Right, inherited from the mother.
Correct. And this mtDNA is particularly vulnerable to damage because it lacks some of the robust repair mechanisms of nuclear DNA. Allotopic expression is a strategy to deal with this.
How does it work?
You basically take the genetic instructions for critical mitochondrial proteins out of the vulnerable mtDNA, recode them slightly so they work in the nucleus, and then insert those genes into the nuclear DNA.
So the nucleus makes the mitochondrial parts instead.
Yes. The nucleus, with its better protection and repair systems, makes the protein, which is then tagged and guided specifically back into the mitochondria where it's needed. It bypasses the damaged mtDNA blueprint.
That's ingenious. Is it being used?
It's being explored primarily for rare genetic mitochondrial diseases, like certain forms of blindness, LHON, or muscle disorders, but the potential for broader anti-aging applications is definitely there.
And there's even talk of mitochondrial transplants.
Yes, that's another cutting-edge area, literally taking healthy mitochondria, perhaps from donor cells or even younger versions of your own cells, and transplanting them into damaged cells. Early research in areas like cardiac repair and neuroprotection is looking into this. Still a very young field, though.
It really underscores how central energy production is to seeing young.
Absolutely. Mitochondrial health is deeply tied to almost everything: immune function, inflammation control, preventing neurodegeneration, metabolic health. It's fundamental.
Okay, so we've covered DNA repair, epigenetic resets, zombie cell clearance, mitochondrial fixes. What about the cell's general cleanup crew? Tell us about autophagy and caloric restriction mimetics, CRMs.
Autophagy, yes, it literally means self-eating.
Yeah.
It's the cell's fundamental quality control and recycling system.
How does it work?
It basically engulfs damaged proteins, worn-out organelles like old mitochondria and other cellular debris, breaks them down in lysosomes, and recycles the raw materials. It's essential house cleaning.
And this slows down with age.
Significantly, yes. The efficiency of autophagy declines, leading to a buildup of cellular junk, which contributes to dysfunction and aging.
So boosting autophagy could be beneficial for longevity.
Definitely. And we know how to activate it naturally. The most potent activator is caloric restriction, CR.
Eating significantly less food.
Right. Reducing calorie intake by maybe 20–40% without causing malnutrition. CR robustly extends lifespan and health span in many species, from yeast to monkeys, delaying age-related diseases. Even shorter periods of fasting, like intermittent fasting, for example, 16 hours without food, or periodic longer fasts, strongly induce autophagy.
But long-term CR or frequent fasting isn't easy or practical for everyone.
Exactly. It can be difficult to sustain, socially challenging, and potentially risky for some individuals. That's where the idea of caloric restriction mimetics, CRMs, comes in. These are molecules that aim to trick the cells into activating those same beneficial pathways as CR or fasting without actually restricting calories.
Fasting in a pill, essentially.
That's the concept, yes. They work by modulating key cellular signaling pathways involved in nutrient sensing and growth. Primarily, they often activate AMPK, an energy sensor, activate sirtuins, longevity-linked enzymes, and/or inhibit mTOR.
mTOR, what's that?
mTOR is a master regulator of cell growth and metabolism. When nutrients are abundant, mTOR is active, promoting growth. When nutrients are scarce, like during fasting, mTOR is inhibited, which switches the cell towards conservation, stress resistance, and crucially, autophagy.
So CRMs try to inhibit mTOR or activate AMPK sirtuins.
Exactly, mimicking the cellular state of fasting.
Are there any specific CRMs showing real promise?
Yes, several are generating a lot of excitement. Probably the most studied is rapamycin. It was discovered in soil from Easter Island, believe it or not.
Wow. And it works how?
It's a potent inhibitor of mTOR. And in lab animals like mice, rapamycin has shown remarkable effects extending lifespan significantly, even when started late in life, improving immune function, reducing cancer risk.
Any downsides?
Yes, it can have side effects, like impacting glucose metabolism or causing mouth sores, which is why research is exploring intermittent dosing strategies, maybe taking it just once a week.
Okay, who else is in the CRM club?
Metformin. This is a super common, widely used drug for type 2 diabetes.
Right, millions take it already.
And interestingly, large studies of diabetics suggest those taking metformin tend to have lower rates of cancer, cardiovascular disease, cognitive decline, and may even live longer than diabetics not taking it, and sometimes even longer than non-diabetics.
How does it work?
It primarily activates AMPK, and has some mild effects on mitochondria and inflammation. Because it's generally safe and well understood, it's the focus of the TAMIN trial targeting aging with metformin.
That's the big one, trying to prove a drug can target aging itself.
Exactly. It's the first large-scale human trial designed to see if metformin can delay the onset of multiple age-related diseases in non-diabetics. If successful, it could be a landmark moment, potentially leading the FDA to recognize aging as a treatable condition.
Huge implications. Any others?
Resveratrol, found in red wine grapes, activates sirtuins. Spermidine, found in things like aged cheese and mushrooms, is a potent autophagy inducer. Both have shown promise in animal models. And autophagy is particularly important for brain health. It helps clear out those toxic protein aggregates associated with Alzheimer's and Parkinson's.
So, keeping the cellular cleanup crew working is vital.
Absolutely. Youth favors growth, high MTOR. Aging often involves neglecting maintenance. Longevity strategies aim to restore that balance, boosting cleanup and repair. Fun fact, very long-lived species like naked mole rats show naturally enhanced autophagy throughout their lives.
So, for listeners, actionable steps here are things like trying intermittent fasting, maybe a 16.8 window.
Yes, or exploring quarterly fasting mimicking diets. Discussing metformin or rapamycin with a knowledgeable doctor, especially if you have metabolic concerns. Eating foods rich in spermidine or resveratrol. Even high-intensity interval training, HIIT, can boost autophagy. And maybe just avoid constant snacking to give your cells that cleanup window.
That's a fantastic overview of the cellular repair toolkit. Now, let's shift gears again, moving beyond just repairing what's there to actually regenerating whole systems. This feels even more sci-fi. Let's start with stem cell-based organ renewal.
Right, this is where we move firmly into regenerative medicine. Stem cells are, as you said, the body's original internal repair system. They're undifferentiated cells that can divide and turn into various specialized cell types to replenish tissues.
But this ability declines with age.
Yes, that's the hallmark known as stem cell exhaustion. Our pools of stem cells diminish, and the remaining ones become less functional. This directly contributes to things like muscle wasting, sarcopenia, slower wound healing, neurodegeneration, and a weakened immune system.
So the idea is to replenish or reactivate these stem cells.
Exactly, to restore the body's innate regenerative capacity, effectively turning back the clock on tissue function. And we're seeing real breakthroughs here. Hematopoietic stem cell transplants, HSCT, which involve transplanting blood-forming stem cells, are already standard treatment for blood cancers and some immune disorders.
But they're being explored for broader anti-aging.
Yes, for things like reversing autoimmune diseases or completely resetting an aged immune system after chemotherapy, a sort of immune rejuvenation.
What about other types of stem cells?
Mesenchymal stem cells, MSCs, are getting a lot of attention. They can be sourced from bone marrow, fat tissue, or umbilical cords. They have strong regenerative and anti-inflammatory properties.
What are they being used for?
Clinical trials are testing MSCs for osteoarthritis, repairing cartilage, heart failure, repairing cardiac tissue, lung diseases, and even neuroinflammatory conditions like MS or Parkinson's. Early results show promise in improving function and reducing inflammation.
And using stem cells to repair the brain directly.
That's a major frontier. Neurogenesis, the creation of new neurons, declines sharply with age. Researchers are using stem cells, often derived from iPSCs, to try and replace lost neurons in Parkinson's disease.
Have they tried this in people?
Yes, there have been early trials. For instance, in 2023, reports emerged from Japan of patients receiving lab-grown dopaminergic neurons derived from iPSCs, showing initial signs of motor function improvement. Similar approaches are being explored for Alzheimer's and spinal cord injury.
Vision too.
Yes. iPSCs are being used to regenerate the retinal pigment epithelium, a cell layer crucial for vision that degenerates in age-related macular degeneration, AMD. Early trials have shown they can halt or even partially reverse vision loss in some AMD patients. The really ambitious long-term goal is stem cell-seeded bioartificial organs.
Growing whole organs outside the body.
Essentially, yes. Using a biocompatible scaffold like a 3D structure shaped like the desired organ and seeding it with the patient's own stem cells. These cells then grow and differentiate within the scaffold, potentially forming a functional organ like a liver, kidney, or trachea.
Grown from your own cells, so no rejection risk.
That's the huge advantage, eliminating transplant wait lists and the need for lifelong immunosuppression.
But